The heart is a driving force for the circulation of blood, which is essential for life. Located in the thorax, just left of the body's midline, this vital organ weighs less than 1 pound. Made up of cardiac muscle, the heart contracts rhythmically throughout your lifetime. In 70 years, it will beat more than 2.5 billion times. The human heart circulates the body's 12 pints of blood every 90 seconds, pumping over 50 million gallons of blood within an average lifespan. Our specimen for study is a pig heart, a mammalian heart resembling man's. Presenting today's dissection is Dr. Tom Quinn, Associate Professor of Anatomy, Creighton University School of Medicine.
Today, we will dissect the heart and attempt to explain its function through a thorough knowledge of its form. By observing the relations of the heart in the thoracic cavity of the fetal pig, you can see that it is located between the lungs in an area called the mediastinum, literally the place in the middle. It is surrounded by a sac called the pericardial sac. The word pericardium comes from the Latin word meaning around the heart. This structure has a strong outer fiber network and an inner layer of smooth cells that glides over an equally smooth layer of cells on the surface of the heart. This layer is called the epicardium. A small amount of fluid is normally found between these layers. For further orientation, turn to a model of the human heart and note the position of the great vessels at this time. These include the largest artery of the body, the aorta, the superior vena cava, and the inferior vena cava, the largest veins, and the pulmonary trunk, which carries blood to the lungs to be oxygenated. Note again the position of the aorta as it arches from the left side of the heart, remembering that the specimen's left is on the right side of your screen. The position of the vena cava, which empties into the right side of the heart. The pulmonary veins return oxygenated blood to the left side of the heart. They can be seen at the base of the heart, that is, the posterior surface. The great vessels are often damaged when the heart is removed from an animal, but there may be some remnant of them on your specimen. Also, observe the right and left coronary arteries. These supply the heart muscle, or myocardium, with blood. The veins that drain the myocardium end in the large coronary sinus, which in turn empties into the right side of the heart. You should now place the heart specimen in front of you. If it has a pericardial sac, you should remove it at this point. Note that it has an outer fibrous layer and an inner serous layer. You will see that the surface of the heart, again called the epicardium, is smooth and glistening. The bulk of the heart consists of the cardiac muscle, the myocardium. There are 4 chambers in the mammalian heart. The external walls of these chambers should be explored before you begin to dissect the specimen. The 2 atria and their small appendages, the auricles, Latin for little ears, and the right and left ventricles may now be seen. Observe that the external border between the ventricles and the atria is formed by the coronary sinus and coronary sulcus or groove posteriorly, and the division between the 2 ventricles is marked by the course of the anterior branch of the left coronary artery. There are, of course, further divisions present on the interior of the heart. We will now confirm this by dissecting the internal structures of the heart. Place the heart specimen in front of you with the apex facing you. Take note of the anterior branch of the left coronary artery. Now make a cut on either side of the artery, as I have done here. This will expose the right and the left ventricles. The word ventricle comes from the Latin word meaning little belly. The ventricles are, in fact, little cavities, as you are now able to see. You also see that the two ventricles are separated by the interventricular septum. The interior of each ventricle has a characteristic texture created by the presence of trabeculi carnii. This term means fleshy beams. We also see portions of the atrioventricular valves, including the chordae tendinii and the cusps, or leaflets of the valves. Observe that there are two parts to the left valve. This valve is also called the mitral valve because the valve resembles the two-part hat that bishops sometimes wear, called a mitre. The right atrioventricular valve, seen here, is called the tricuspid valve because it has 3 points. Once more, observe that the valves are anchored to the chordae tendinii and that these, in turn, are attached to small papillary muscles extending from the myocardium of the heart wall. Turn your attention once more to the model of the heart. The valves you have just seen in the heart dissection are called atrioventricular valves. As their name indicates, they are placed between the ventricles and the other chambers of the heart, the atria. When the heart contracts during systole, the blood in the ventricles is forced against the undersurface of these valves, and they close. This prevents blood from going back into the atria from which it came. The blood is instead forced to leave the left ventricle by way of the aorta and the right ventricle by way of the pulmonary trunk. When the heart relaxes again, it is said to be in diastole. Blood then flows from atria, plural for atrium, an entryway, into the ventricles. Each atrium also has a small side chamber called the auricle, meaning ear. We will now examine these chambers in the heart dissection. Divide the upper portion of the heart, as I have done. You can see that the interior of the atrium is quite smooth. The auricles, however, have a texture that is similar to the ventricles. This is caused by the pectinate muscle. This term is applied because of the resemblance of these muscles to a comb called pecten in Latin. Reaffirm that the atria are in communication with ventricles via the atrioventricular valves on each side. You should now open the aorta and the pulmonary trunk in this fashion in order to expose the semilunar valves. These relatively small valves are named for their half-moon shape. Each of the valves has 3 of these half-moon cup-like portions. When blood has been pumped into the great vessels, these valves prevent it from returning to the ventricles. This saves a great amount of effort for the heart. It is important to try at this time to find the openings of the coronary arteries. They should be found in the semilunar valves of the aorta. The right coronary artery has been divided in this specimen in order that a probe could be inserted into the opening. We see the probe in the opening at this point, just superior to the aortic semilunar valve. A review of the anatomy of the heart should always be carried out with the function of the heart in mind. Blood that has become deoxygenated flows from other veins into the superior and inferior vena cava, as well as the coronary sinus. This blood is then collected in the right atrium. Passes through the right atrioventricular valve, the tricuspid valve, and into the right ventricle. When the heart contracts in systole, this blood rushes against the tricuspid valve and closes it. The blood then must choose the path of least resistance, the pulmonary trunk. The valve of the pulmonary trunk, one of the semilunar valves, keeps blood from rushing back into the heart. The blood in the pulmonary trunk flows into the circulation of the lungs, where it is oxygenated. This oxygen-laden blood flows by way of the pulmonary veins into the left atrium. It then flows into the left ventricle through the mitral valve during diastole, or heart relaxation. When the ventricle contracts once more, blood enters the aorta. Backflow again is prevented by a semilunar valve. This concludes the dissection and anatomy of the heart.
The most important muscle of the body is the heart, yet it is only about the size of your fist. This powerful organ pumps blood through a series of chambers and valves into the cardiovascular system. Our investigation has highlighted the working operation of the mammalian heart and has identified the anatomical as well as the physiological functions of this vital organ.